We created organisms that could turn starlight into unlimited clean energy. For a few weeks, it worked perfectly. Then the organisms started reproducing faster than predicted, then mutating, then spreading beyond containment. And by the time we understood what was happening, the thing we built to save Earth was consuming it instead. This isn't just science fiction. The biology is real. And once you create a self-replicating organism optimized for survival, you can never fully control what it becomes. And it doesn't fail the way you'd expect. At first, the energy production exceeds all projections. The organisms behave exactly as designed. Humanity celebrates. Then the reproduction rate accelerates. Containment becomes difficult. The organisms begin adapting to countermeasures. And eventually, the thing you created to feed on stars starts feeding on Earth instead. Let's start with why this solution seems perfect and why that's exactly the problem. Right now, Earth is dying. The sun is dimming because of an alien microorganism called Astrophage, a single-celled organism that reproduces by consuming stellar energy. Without intervention, global temperatures will drop by 10 degrees Celsius, crops will fail, billions will die. But then we discover something remarkable. Astrophage isn't just consuming the sun's energy, it's converting it with near-perfect efficiency. 1 gram of astrophage contains more stored energy than a kilogram of uranium. In Project Hail Mary, astronaut Ryland Grace and his alien partner Rocky realized they can weaponize this, breed astrophage in controlled conditions, harvest the energy, and use it to save Earth. It's elegant, it's efficient, and the math works perfectly. The energy density is extraordinary. Astrophage reproduces by mitosis, splitting into 2 identical copies, using only starlight as fuel. No waste products, no pollution, just pure energy generation with exponential growth potential. In theory, you could power an entire city with a few kilograms. In theory, you could eliminate fossil fuels within a decade. And then Reactor 1 goes online. The containment vessel walls glow with stored energy, more power than a nuclear plant, contained in a space the size of a swimming pool. Within 48 hours, rolling blackouts across the eastern seaboard end, permanently. By week 2, global energy prices drop by 40%. Scientists appear on news broadcasts calling it the greatest achievement in human history. World leaders announce plans to decommission coal plants within the year. For the first time since the Industrial Revolution, humanity has found a perfect energy source. The celebrations last 3 weeks. But here's the thing about organisms that reproduce exponentially. They're not machines. They evolve. And evolution doesn't care about your intentions. Which brings us to the first sign that something's wrong. Let's say you're 3 weeks into large-scale astrophage cultivation. You're managing a facility with trillions of organisms, all breeding inside sealed photobioreactors. Every metric looks perfect. Then you notice the reproduction rate has increased by 0.3%. Not much. Barely outside the margin of error, but it's consistent across all reactors. You run the numbers. At current rates, you'll hit your energy targets 2 weeks ahead of schedule. The project director calls it a win, but you keep staring at that 0.3%. You start checking the monitors twice before leaving at night. You don't mention it to anyone. The reactor room feels warmer than it did last week. Not enough to trigger alarms, but enough that you notice. Because exponential growth doesn't work linearly. A 0.3% increase today becomes 0.6% next week, 1.2% the week after. Small deviations compound faster than you can model them. In biology, unexpected reproduction rate increases usually mean one of 2 things. Environmental conditions are more favorable than predicted, or the organism is adapting. With trillions of astrophage cells undergoing billions of cell divisions, random mutations occur constantly. Most die. But a few, the ones that reproduce faster, resist temperature extremes, breach containment materials, those survive. You're not just breeding astrophage, you're creating evolutionary pressure. And evolution is optimizing for one thing: survival. By week 5, the reproduction rate is 2% above baseline. Containment protocols need updating twice. You increase the frequency of genetic sequencing. The results show minor variations. Nothing alarming, probably. And this is what most people miss about biological systems. They're not engineering projects. You can't debug biology the way you debug code. A bacterium doesn't follow a blueprint. It follows one rule: survive and reproduce. And then you get the call that primary containment has been breached. Around week 8, alarms start going off in Reactor 7. Astrophage has breached the inner containment vessel, not through mechanical failure. The seals are intact, the pressure is stable, but Astrophage is on the other side of a barrier that should be impermeable. You seal the secondary containment. The organisms are still spreading. You flood the chamber with UV radiation, which should kill them instantly. It slows them down, but within hours they're reproducing again. And then you realize what's happening. They're adapting in real time. Through the observation window, you can see it. Black colonies spreading through the transparent cooling pipes, places that should be sterile. The UV chambers glow with maximum intensity, but the colonies remain, pulsing faintly, almost imperceptibly expanding, growth where nothing should survive. Astrophage reproduces every few hours. That means thousands of generations are passing in the time it takes you to run one round of tests. With trillions of organisms and constant selection pressure from your countermeasures, evolution is happening at a speed you've never seen in nature. Studies of antibiotic-resistant bacteria show this exact pattern. Expose a population to a toxin that kills 99.9% of them, and the surviving 0.1% become the parents of the next generation. Within weeks, you have a population that's completely resistant. With Astrophage's reproduction speed, you're seeing bacterial evolution compressed into days. Here's what makes this particularly concerning. Every containment breach creates stronger organisms. You're not fighting the original Astrophage anymore. You're fighting its descendants, the ones that survived your best defenses. By week 10, Astrophage has spread to 3 facilities. Genetic sequencing shows it's no longer identical to the original strain. Some variants can survive in lower light conditions. Some have developed resistance to chemical sterilization. Some are reproducing in environments that should be lethal. You implement emergency protocols, full facility lockdown, atmospheric isolation, but the breach reports keep coming. And then you detect Astrophage in the water supply. Even if you could re-establish containment, even if you could track every escaped organism, Astrophage has another problem that might make extinction inevitable. It's no longer feeding only on starlight. Week 12, you're monitoring satellite data when you see it. Energy signatures dropping across the northeastern power grid. Not mechanical failures, not cyber attacks. The solar panels themselves are losing efficiency. You send teams to investigate. They find Astrophage colonies on every panel, not just feeding on sunlight, feeding on the electrical current in the panels themselves. The organisms have evolved to consume any available energy source. Energy, to an organism, is just chemical bonds that can be broken. Astrophage evolved to harvest stellar radiation, but radiation and electricity are both electromagnetic energy. A mutation that allows Astrophage to metabolize electrical current isn't exotic, it's inevitable. And once organisms evolve to harvest electrical energy, the jump to living tissue isn't as impossible as it sounds. The energy molecule inside every cell in your body stores power the same way a battery does, by moving electrons between chemical bonds. Within weeks, reports come in from agricultural zones. Crops are dying. The plants themselves are being consumed. Astrophage has learned to metabolize organic matter. This is exactly what happened when invasive species spread across Earth. Cane toads in Australia, kudzu in the American South, zebra mussels in the Great Lakes. You introduce an organism to a new environment with no natural predators, and it doesn't just survive, it dominates. Except Astrophage isn't competing with other species for resources. It's consuming the resources themselves. You realize what you're seeing, an organism that can feed on sunlight, electricity, and biological tissue. An organism that reproduces exponentially. An organism that's spread across 6 continents in less than 3 months. You've created the perfect invasive species. And it's no longer a question of containment. It's a question of whether Earth's ecosystems can survive it. Month 4. This is your reality. You're standing in the situation room, watching red zones spread across the global map. Every sterilization attempt creates selection pressure. The survivors come back stronger. You step outside and the air tastes different, metallic. The sky has a faint greenish tint. Astrophage colonies suspended in the upper atmosphere, feeding on solar radiation before it reaches the ground. And the worst part, you can't remember what normal used to feel like. Was the sun always this dim? Were the nights always this dark? You know, with absolute certainty that you did this. You helped breed the organisms. You celebrated when they exceeded projections. You ignored the 0.3% increase. The thing you created to save 8 billion people is now killing them, and there's nothing you can do to stop it. So what's the solution? The obvious answer is sterilization, gamma radiation, extreme heat, chemical agents, something that kills every astrophage cell on Earth simultaneously. Problem solved. Except you'd have to sterilize the entire biosphere, the atmosphere, the oceans, the soil, and astrophage is already in all of them. Even if you killed 99.99% of the organisms, the surviving 0.01% would be the most radiation-resistant, heat-tolerant, chemically immune variants, and they'd repopulate within weeks. It's theoretically possible, but it would require sterilizing Earth so thoroughly that you'd kill most other life in the process. The other solution is a genetic kill switch. Engineer a mutation that causes astrophage to stop reproducing or self-destruct, release it into the population, and let it spread. And that might work if you were dealing with the original organism. But astrophage has already mutated past your baseline genetic code. The population is now so genetically diverse that any kill switch would only affect a fraction of them. Right now, we don't have a proven way to eliminate a self-replicating organism once it achieves global distribution. And that's not a small problem. It's the problem with any technology that tries to solve a crisis by creating something that can reproduce independently. We have theories, we have models, but we've never faced an invasive species that can adapt this fast, spread this far, and consume this much. And this changes how we think about biological solutions to existential problems. Because Project Hail Mary makes the astrophage solution look like a triumph, humanity's ingenuity saving the day. In reality, it would be the last mistake we ever make. So when we ask whether we could solve an energy crisis by breeding star-eating organisms, whether we could actually implement Project Hail Mary's solution, maybe we're asking the wrong question. The question isn't whether we can breed organisms that harvest stellar energy. We probably can. The biology is sound. The energy density is real. The question is whether we can control what happens after. Because self-replicating organisms follow one rule: survive. And the moment you create something optimized for survival, you're creating something that will eventually optimize for survival at your expense. Maybe we'll figure out containment. Maybe we'll develop genetic safeguards that can't be mutated around. Maybe we'll find a way to make biological solutions safe. But Project Hail Mary's astrophage solution isn't just a risky gamble. It's a test of whether we can create life and remain in control of it. And here's what we keep learning. Evolution never accepts good enough. It keeps optimizing until something dies. And when you're competing with something that reproduces 1,000,000 times faster than you do, you're not the one who gets to survive that optimization.